Hello, my name is Rob Hutchison, and I'm the Chairman and CEO of BiOasis. BiOasis is not your typical pharmaceutical company, as it does not develop new drugs per se, but rather we take already existing FDA-approved therapeutics and/or experimental drugs from drug companies and link these drugs through our patented proprietary technology to a human protein that acts as a vector to shuttle these therapeutics across the blood-brain barrier. More on this later. This slide is courtesy of our lawyer. BiOasis technology was discovered and initially underwent scientific study at the University of British Columbia, Vancouver, BC. BiOasis acquired this technology in 2007 through the issuance of shares in BiOasis to the university. We then took BiOasis public by way of a reverse merger into a TSX Venture-listed company. To date, we have raised slightly over $6.5 million to ready the technology for commercialization. As I mentioned, the technology was developed to enable therapeutic compounds to be delivered into brain tissue. The human brain has a blood capillary network, imaged to your right, that is over 450 kilometers long and is the largest blood capillary network in the human body. Within this network, we find the blood-brain barrier. This barrier protects the brain from toxic compounds entering the brain tissue, and it does a wonderful job at it. It does it so well that it blocks over 95% of all potentially beneficial therapeutic compounds from entering the brain. Cancers, for example, can be effectively treated throughout the body with existing therapeutic compounds, but they have little to no effect on treating cancers within the brain for the central reason that they can't cross out of the capillary network and through the blood-brain barrier and get into the brain tissue to fight the disease. This is a huge unmet medical need. So to overcome this problem and potentially find a solution to meet this unmet medical need, BiOasis developed Transcend. Our human protein-based vector has been shown by us and independent third parties that Transcend can shuttle both small and large therapeutic compounds across the blood-brain barrier and deliver those compounds into the brain tissue. The key to Transcend's effectiveness is a process called receptor-mediated transcytosis. Transcend binds to receptors found on the walls of the capillary network that actually pulls Transcend and the compounds linked to it across the blood-brain barrier and into the brain tissue. So through Transcend, BiOasis has the potential to deal with one of the greatest challenges in treating brain diseases and disorders, the ability to deliver therapeutics into the brain tissue. We have and continue to protect our assets through patent strategies and know-how. Our development programs are quick and very efficient, all backed by proven and independently validated science and a strong and experienced leadership team. Our business models are designed to work with industry as opposed to against it. We are developing a number of in-house programs that we are actively pursuing, along with making Transcend available under license for others to use to exploit the capabilities of Transcend. As I mentioned, Transcend has undergone extensive testing and independent validation. In 2002, the Southern Research Institute in Alabama tested the delivery of a cancer therapeutic, doxorubicin, in animals who had tumors implanted in their brains. They showed two significant findings. One, an increase in overall survival rate of the animals who received the Transcend-linked doxorubicin, and two, tumor shrinkage, and in a number of subject animals, complete eradication of the implanted tumors. In 2003, BioMarin Pharmaceuticals successfully delivered an enzyme that is used to treat a lysosomal storage disease into the brains of subject animals. And in 2007, the University of Alabama delivered an adenovirus across the blood-brain barrier and into the brain tissue. These are very large viruses and have been studied in the use of gene therapy. So in taking what others have shown, BiOasis in 2010, working with the National Research Council of Canada, labeled Transcend with a fluorescent dye to yet again confirm transport into the brain tissue, but also to look for distribution with the brain cells themselves, an important step in understanding where the compounds would localize within the brain tissue. With knowledge in hand, we looked at addressing a very large unmet medical need in dealing with a form of breast cancer, that in over 30% of cases result in the onset of brain metastases, which often leads to death. So in 2011, we showed that Transcend could deliver the drug Herceptin that is used to fight this form of breast cancer across the blood-brain barrier. At the same time, in working with the British Columbia Cancer Centre, We tested the effectiveness of Transcend-linked Herceptin compound, an entity we now call BT2111, against Herceptin cell lines, and to our delight, with an unexpected result, the new compound BT2111 killed 3 times the number of cancer cells than Herceptin did on its own. With each of the tests coming back positive, we advanced the BT2111 program forward in earnest. The next step in the overall development of the program was very critical to us. We had to show that BT2111 remained cytotoxic and invoked the human immune system to respond. We showed this in an ADCC assay, which is a mouse model to predict activity in the human body. The results confirmed that BT2111 remained cytotoxic when introduced to human donor immune system cells, exactly what we were hoping for. So with the science and testing all showing great promise, let's turn to the opportunity. The overall market for neurological drugs, or what is also referred to as the central nervous system diseases drug market, is estimated to be approaching $100 billion annually. The chart shown here shows the top CNS therapeutic areas. It is the second largest drug market only behind cancer, yet it is hampered by the ability to get therapeutics across the blood-brain barrier. This is very evident when you look at the cancer numbers in this chart. By opening up the opportunity for the blood-brain barrier transport, you would expect significant changes in the numbers in this chart going forward. One of our main goals at BiOasis is to limit the risk for our stakeholders, which is why we are and will remain focused on working with therapeutics that are already approved, such as is the case with Herceptin. In the programs we have underway and those we are considering, we also look to remove risk by assessing things such as market size and patent life of these therapeutics. In the case of Herceptin, it comes off patent in the United States of America in 2014 and 2015 in other jurisdictions. Roche's annual sales for Herceptin are over $5 billion. And typically, once a drug comes off patent, the generic versions hit the market, and the original drug manufacturer, in this case, loses as much as up to 2/3 of their annual sales revenue. So as part of our de-risking process, we look to opportunities to potentially extend a drug manufacturer's patent protection. BT2111 is classified as an NCE, or a new chemical entity, and therefore one can obtain new patents around the NCC given certain circumstances. Since we showed A, brain transport, and B, an increased cancer killing effect, BT2111 could provide certain patent protection going forward to a pharmaceutical partner. So I've talked about the overall opportunity and our development/business models. So let's look a bit closer at our internal programs to which we are focusing on. We chose these internal programs for a number of reasons, such as unmet medical need, but two as well, where we had valuable patents or patent-pending applications. In the area of lysosomal storage disease, off the work done in 2003, we have recently entered into a collaboration with Shire HGT, one of the largest specialty pharmaceutical companies in the world. Shire's focuses on rare diseases, and LSDs is one of their main focuses. Shire and others whom target the LSD market have been hampered with the treatment of LSDs that target the brain. Same old problem, the blood-brain barrier. We are working with Shire to hopefully overcome that problem. I've mentioned BT2111, and we are very excited about the findings to date. We are now working with Dr. Quinton Smith's lab at Texas Tech University, where under Quinton's leadership, we are working with his renowned brain metastases animal model. With solid data from this work over the coming months, we will enter a very exciting period that may include advancing the development of BT2111 with a pharmaceutical partner. BT2112 is a combination of a cancer drug called Erbitux and Transcend. Like the Herceptin combination, BT2112 is targeted at the potential treatment of brain cancers that develop in people suffering from primary cancer sites elsewhere in the body. In assessing the revenue opportunities for our internal programs, the chart on this slide shows 3 drugs targeting the LSD market that could significantly benefit from brain transport. Our revenue model would be to obtain an upfront license fee, milestone payments, and then an ongoing royalty on annual sales. As these rates vary, we do not want to speculate in this presentation as we might undervalue the value of our technology at this time. In the area of brain cancer, again for these indications, there would be license fees would fall in line with industry standards and royalty payments. As patients in both of these indications have a 30+% risk to develop brain cancer, and where the treatment in brain would be the indicated drug, brain transport becomes key. It would make sense that physicians treating patients with HER2-positive breast cancer, for example, would treat those patients with BT2111 should it obtain FDA approval. The same would potentially hold true for Erbitux. I've added this slide to give you a visual representation of our BT21 brain transport findings. The image was taken by a confocal microscope and is a slice of a subject animal's brain. The brain capillaries here are shown in red, and the blue dots are the nuclei of the brain cell. The green dots are BT2111. So you can clearly see that BT2111 has come out of the capillaries, crossed the blood-brain barrier, and are now localized within the brain tissue. If you were to compare this to Herceptin on its own, you would see less than 1/10 of the amount in brain tissue. Most of it is stuck in the capillaries. Aside from the internal programs that I have described in the prior slides, we are actively seeking licensing agreements with companies whom have either experimental drugs they'd like to target to the brain or have blood-brain barrier transport desires. These agreements typically start as a collaboration to provide a proof of concept that will lead then to a license provided by BiOasis to the third party under industry standard terms. BiOasis is managed by a board of directors and management team with strong industry experience. Their knowledge and relationships will enable timely development of our products and the structuring of profitable agreements with industry partners. It is supported by very experienced intellectual property attorneys at Cooley Law Group, whom are working with us to ensure that the company has strong competitive advantage through its patent portfolio. This slide shows the current capitalization of BiOasis as at February the 29th, 2012. I would like to thank you for personally reviewing this presentation, and if you have any further questions, please don't hesitate to get a hold of me at the number listed below. Thank you.